Okay, thanks very much, everybody, for giving me a chance to talk today. I'm going to talk about a quantitative neutralizing antibody lateral flow point of care assay for measuring neutralizing antibodies against SARS-CoV-2. And just up front, this is an important collaboration between my team and some others at the Doherty Institute and David Anderson and his team at the Burnett Institute. It's funded by the Victorian State Government and also an ARC Centre of Excellence. And as a disclosure, there's a provisional patent covering this test and the underlying technology that has been submitted through the University of Melbourne. So just as a very gentle beginning, and I'm sure I don't really need to say this, but just to make the point, here's the SARS-CoV-2 virus, here's a cartoon of the virus, and everyone recognises these characteristic spikes. And the spikes how the virus binds to our cells, and they bind via the ACE2 receptor on our cells. And the spikes have at their tip what we call the receptor binding domain, which is essentially like a key that unlocks the door to ACE2 and therefore allows the virus to get into our cells. And when we're infected with this virus, or when we inject a vaccine, we make antibodies against all parts of the virus or the spike. And it's these antibodies against the RBD part of the virus that appear to be the most important for blocking the virus from binding to ACE2 and therefore blocking infection. So it's been shown that approximately 90% or more of neutralizing antibodies in COVID patients target the receptor binding domain. And if you remove those, you remove most of the neutralizing antibody from sera, from patients. So this knowledge has allowed has helped us to understand personally how the virus infects cells, but also how to develop some assays where we can measure neutralizing antibodies. And the first assay that came out that exploited this understanding was a study from Lin-Far Wang and colleagues published last year in Nature Biotechnology, where they used the RBD ACE2 interaction as an ELISA-based inhibition assay to measure neutralizing antibodies. So I'll just briefly explain what they did. And to begin with, I'll show you, this is just your conventional virus neutralization test where you infect cells typically in a high containment laboratory. The virus will infect the cells, and if you have antibodies present in a patient's sera, they can bind to these viruses and stop the virus from infecting cells. This is a really powerful assay. It's one of the gold standard type assays, but it takes about 3 days for the infection and the readouts to take place, and it has to be carried out in a high containment laboratory, or at least a mid-level containment laboratory if you use a pseudovirus. So that's not great if you need quick results. And so this is where Lin-Far Wang and his team developed this ELISA inhibition type assay called the surrogate virus neutralization test. And in this case, they've made a soluble RBD protein and they've conjugated it to horseradish peroxidase. And on the ELISA plate, they've got the ACE2 receptor again as a protein just bound to the plate. So the RBD will bind to the ACE2 and the horseradish peroxidase will cause a change in the color when you add substrate. So you get a clear color readout when RBD binds to ACE2. But then if you add neutralizing antibodies from patient sera, for example, they can stop the RBD binding to ACE2 and you lose the color. So you get a readout in the form of inhibition of RBD ACE2 interaction. And in that paper, they showed that if they look at in a cohort of patients who've had SARS-CoV-2 in Singapore, they can see a clear range in inhibition ranging from about 20% up to about 100% inhibition of SARS-CoV, of the RBD ACE2 binding compared to a control cohort where they never saw more than 20% inhibition. So that's a fantastic assay. This has taken, this has basically resulted in an assay that can take place in about 3 hours rather than about 3 days. And it can be performed in a lower containment level laboratory, but it still requires a laboratory and ELISA plate readers and the like. So we wanted to see if we could take that kind of an idea and turn it into a point of care test in the form of a lateral flow assay. And I'm sure you've all seen these lateral flow assays. They're used for various things, pregnancy tests and other sorts of tests. And there are also some lateral flow assays that measure antibodies against SARS-CoV-2 virus antigens. Here's a picture of an example here. You typically have a control line and a test line. So we tried to develop a test like this that uses the RBD ACE2 interaction as the readout. And the idea is that the antibodies in sera from patients or from vaccinees will block that interaction, and we'll see the line become weaker if there's neutralizing antibodies. So just to show in a cartoon form what we're trying to do, we have a nitrocellulose strip and we bind human ACE2 to the strip. And then we take RBD from the SARS-CoV-2 spike, and it's a biotinolated RBD, and then we have an antibiotin gold antibody, and it's the gold that gives you the colored readout on these assays. So if you add the RBD gold to this strip, you'll get, obviously the RBD will bind the ACE2 and you will see a red signal. But if you add antibodies that can block that interaction, that stops the RBD binding ACE2 and you lose the red signal. An advantage of this assay, like the assay Lin Fa Wang developed, is that it doesn't matter what antibody type you're looking at. It could be IgG or IgM or IgA, and it doesn't even matter if it's a human or another species, macaques or mouse or whatever. It just requires that there's some protein there that will block this interaction. So the idea is if you get a strong red line, that means there isn't any neutralizing antibody. If you get a faint red line, that means that you might have a weak neutralizing antibody response. And if you see little or no red line, that means you've got a strong neutralizing response. So here's our first setup that we developed. And this is one of the strips here. If you have you add a sample of blood or serum, and this serum will run across, it will pick up the RBD gold conjugate, and that will run through the action of the wicking pad at the end of the strip. And as it crosses across this ACE2 line, the RBD will bind, setting up a red line, and the serum will continue to flow towards the wicking pad. Now, if you have a COVID positive sample that has antibodies against RBD that can neutralize, when it runs across, it'll pick up the RBD, and the RBD and the antibodies will combine, and these antibodies will stop some of the RBDs from binding to ACE2. And so essentially you have a fainter red line. So we wanted to see if this works with actual neutralizing antibodies that we know were neutralizing. So we took two anti-RBD antibodies that we knew were not neutralizing and two antibodies that we knew were neutralizing, and we tested them in these assays. And we just tried a range of molar ratios of antibody to RBD. So the top two antibodies, they're anti-RBD antibodies, but they are not neutralizing. So it didn't matter what molar ratio we used, it didn't change the intensity of the red band, which represents ACE2 RBD interaction. Now that's what we'd expect because these are not neutralizing antibodies. The next row shows two neutralizing antibodies. And in this case, when the molar ratio got high enough, we saw that the red line disappeared. And you can see, especially with antibody B, it disappeared gradually. So that is a proof of principle that we can use antibodies to block the RBD ACE2 interaction in this assay. Now, this is a semi-quantitative assay by eye. You can see that the line has faded when you've got neutralizing antibodies, but we can get much more accurate readings using this instrument here. It's an Axon strip reader, and that's just the Axon strip reader reading the intensity of the bands. And so the neutralizing antibodies, you can see as the molar ratio increases, we lose the intensity of the red band. So that's showing proof of principle. Now, what about with COVID patient plasma samples, which we know should contain neutralizing antibodies? So here's 10 examples from 10 healthy COVID negative plasma samples. So there's no neutralizing antibodies here as expected because these were non-infected individuals, and the red line is pretty consistent in each of those individuals. Here's 10 COVID positive plasma samples, and you can see now we get quite a range in the intensity of the red line. We have examples where we have very weak or no inhibition of the red line. It's pretty similar to the healthy controls, so that's a weak response. Then we have some that are faint lines. These are intermediate neutralizing antibody responses. And then we have some where there's no red line. These are strong neutralizing antibody responses. And we looked at, in this case, it was about 35 samples. COVID negative samples, they didn't show much inhibition, maybe up to 20% either, or 20% either side of the mean. Whereas the COVID positive samples, we saw inhibition ranging from about 0 to up about 95% inhibition. We included a few samples from individuals who'd been infected with other viruses, including influenza, picorna, and RSV, and they didn't, as expected, have any neutralising antibodies that could block RBD ACE2 interaction. Importantly, we needed to know, how does this relate to a gold standard assay? And so we used a microneutralisation assay where virus is used to infect viro cells, and we measure the degree of infection over the 3-day gold standard microneutralisation assay. And we found there was a very nice correlation between the amount of inhibition by our lateral flow assay and by the microneutralisation antibody titer in the gold standard assay, as you can see here with these black dots. And the controls are all here clustered in amongst the 0 lateral flow assay readout. So we wanted to now make this into a test that was feasible for using in the real world. So we needed to really set it up in a cartridge form, and we also needed some control lines on this. So we set up this test as follows. We have our ACE2 RBD inhibition line, we have a control line or a reference line, and then we also built in a total antibody, a total anti-RBD antibody line, so we can see if somebody has been infected and have anti-RBD antibodies, even if they don't have neutralising antibodies. So the way this strip works now, we have our 3 lines. Now ACE2 bound to the strip here, which is the inhibition line over here. Then we have a control or a reference line, and this is just an irrelevant antigen antibody pair. So in this case, on the strip, we have anti-chicken IgY, and the third line, the total anti-RBD antibody line, here we've just got RBD, the protein itself. So the way it works is in the gold pad where we have the reagents, we have RBD with gold label on it. We have chicken IgY with gold label on it. And of course, we add to our sample pad the patient sample, which may or may not contain anti-RBD antibodies. And as these diffuse along the strip through the flow, the RBD will bind to the ACE2 and you'll get a gold mediated red signal, unless there's anti-RBD antibodies that can neutralize this interaction and reduce the intensity of the signal. The chicken IgY will bind to the anti-chicken IgY, and that won't be interfered with by anything in the serum sample. So that's our standard control and reference line. And then the anti-RBD antibodies, if they bind to RBD with gold on it, then the other arms of the antibodies can bind to the RBD that's immobilized on the strips, giving us a line that just says there's anti-RBD antibodies present in the serum. And so you end up with 3 lines that tell you information about what's in the serum. So here's this prototype cartridge that's been used to look at serum samples from either healthy controls or COVID-19 patient samples. And so you can see the healthy controls, we have a very strong line at the ACE2 RBD line, which is what you'd expect because there's no inhibition, no neutralizing antibodies. The reference line is the same across all of these samples. And there's no sign of anti-RBD antibodies in this healthy controls. Now we look at the COVID patient samples, we see that the inhibition line, it varies, and in some cases it's almost missing, which is indicating that this patient has a strong anti-RBD neutralizing antibody response. The reference line hasn't changed, and some of these patients now show strong anti-RBD total antibodies as well. And that's what you'd expect, a lot of total anti-RBD antibody and a lot of inhibition, for example, here. Sometimes, however, you have total anti-RBDs that are detectable, but very little inhibition. And that's why we need an inhibition type assay to know if someone has neutralizing antibodies. So here's a readout from these samples. We tested 70 samples using this test, and you can see again, we get a range from 0 to 100% inhibition with our assay. And again, these samples, they correlate quite nicely with a gold standard microneutralization test. So just to use this assay to demonstrate a few points, we can, for example, demonstrate waning neutralizing antibody in longitudinal samples from one patient. So here's patient C1, and this patient was sampled over a few different time points after being infected, and you can see the percentage of it, the inhibition line gradually gets darker, which means the neutralizing antibodies are waning, and the total antibody line gets fainter as the total anti-RBD antibodies get diminished, and the control reference line doesn't change. And here's our healthy controls. And that's just a graph to show that over time, the sample from this patient, you gradually lose inhibition. This was also an interesting example. This is a different patient, C2, at their first bleed time point. They have a strong anti-RBD antibody response, but very little inhibition. And again, that's why we need an inhibition assay to know if someone has neutralizing anti-RBD antibodies. Now, for this to really be useful in the real world, we need it to work with blood, not serum, because we don't want to have to separate blood into serum in the lab. So we've set up a version of this where we have an anti-glycophorin A antibody built into the strip that takes the red blood cells out. And using this assay, we can show that if we take plasma from a patient with known neutralizing antibodies, we mix them with red blood cells. So we're, in a sense, we're fabricating blood for this purpose, just to see if the blood interferes with the response. And here's the plasma by itself. We find that this assay worked very well. The red blood cells didn't interfere with the response. We see neutralization that gradually diminishes with time, and the red blood cells in the mix in the upper panel didn't interfere with that response. And this is just a comparison of the plasma with or without red blood cells added, and we saw the comparable results in terms of how much inhibition we measured. And of course, now that people are getting vaccinated, we wanted to make sure that this works with immunized vaccinated samples. Here's a representative 3 samples from an unvaccinated individual. After the prime, this is Pfizer vaccinees, we didn't see much change in the intensity of the total anti-RBD or the amount of inhibition. But after the boost, now you can see there's a very clear total anti-RBD antibody response and very clear evidence of inhibition because this line has disappeared. Interestingly, if they were previously infected, then even just after the prime of the vaccine, we saw a total anti-RBD antibody response that's very clear and very clear evidence of inhibition. And here is measuring these vaccinees, total inhibition versus microneutralization titer. And again, we see a good correlation between our assay and a gold standard microneut assay. And then this is the last data slide. Can this be adapted for variants of concern? And the answer is yes. We've already tested the triple mutant RBD from the beta variant or the South African variant. And so here's the Wuhan variant in the top panel. And you can see, again, these are the same samples I've shown you the last couple of slides. We see clear inhibition for C1 and C1.3 and some inhibition at C1.5. And that's much diminished when the RBD comes from the triple mutant virus. So that's what you'd expect because the triple mutant RBD binds is harder to neutralize with anti-sera from people who've been infected with the Wuhan virus. And this just shows a few different samples that had clear inhibition neutralizing antibodies against the Wuhan strain shown in blue. But when the triple mutant RBD was used, there was a clear reduction in the amount of inhibition, indicating that this assay can be used to test against variants of concern. So just in summary, we have an assay that can be semi-quantitative by eye, and you can get a readout giving you a quantitative readout using this Axon strip reader. So here's a sample that you would call positive. They have greater than 50% inhibition, and here's a negative control sample that you would call negative, and they have less than 50% inhibition, and you can get a very accurate readout from that. So we've developed a point of care assay for neutralizing antibodies that can give a readout in about 20 minutes. This should help with emerging questions about immunity to SARS-CoV-2, for example, following infection or vaccination. And these are really important questions that keep coming up now. Who is immune? How long do they stay immune? And are they immune to the variants of concern? And it just remains to thank the various large numbers of people involved in this work. Funding was from the Department of Health and Human Health Sciences Services, the ARC Centre of Excellence. The work is a collaboration between the Burnett Institute and the Doherty Institute. These people from my team, especially Tom Fulford, Nick Gerard, and Marcin Chulia, and Damien Purcell at the Doherty Institute, and Hoi Van, Xuning Zheng, and David Anderson at the Burnett Institute, and many other people, Heidi Drummer especially, and many others who've been involved in making this work. So with that, I'll stop and thank you very much for your time.